The overall goal of this clean sampling and analysis protocol is to provide adequate techniques for obtaining reliable data for trace metal studies, especially when low detection limits are required.
This method can help answer key questions in environmental chemistry field in aquatic environments with dynamic hydrological and geochemical conditions.
The main advantage of this technique is that under the suggested guideline, reliable data can be obtained and related to variable key environmental parameters.
Though this method can provide techniques to obtain information in rivers and estuaries, it can also be applied to other systems such as lakes, coastal zones, and oceanic environments.
Generally, individuals new to this method will struggle due to the lack of knowledge on contamination controls and the inability to recognize proper means to eliminate contamination during trace metal analysis.
Demonstrating the procedure will be Qiping Li and Peiyu Lin, students from our labs.
To assemble the sampler, connect 4-meter-long fluorinated ethylene propylene tubing to a 1.5-meter chemically resistant silicone pumping tube. Insert a polypropylene Y connector into the pumping tube. Connect a 50-centimeter pumping tube to one outlet and a 0.45 micrometer capsule filter to the other. Assemble the tubings in a clean room after they are cleaned as described in the text protocol, and store the assemblage in 2 layers of polyethylene bags. For sample containers,